What do you understand by organelles? Organelles are subcellular structures within eukaryotic cells that carry out specific functions to maintain the cell's life and metabolism. Examples of organelles include the nucleus, mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes. Each organelle is specialized to perform a specific set of tasks, such as protein synthesis, energy production, and waste removal, that keep the cell functioning properly. Compare the size cells and organelles. Cells are much larger than most organelles, with most eukaryotic cells ranging in size from a few micrometers to tens of micrometers in diameter. Organelles, on the other hand, are typically only a few nanometers to a few micrometers in size. The nucleus, for example, is usually about 10 to 30 micrometers in diameter, while ribosomes, which are the smallest known organelles, are only about 20 to 25 nanometers in size. The size difference between cells and organelles reflects the fact that cells are made up of many different organelles, each with its own specific size and function, working together to maintain the cell's overall health and survival. What are merits of organelles? The presence of organelles in eukaryotic cells has several advantages, including 1. Division of labor. Each organelle performs a specific set of tasks, allowing for a division of labor that enables the cell to be more efficient in carrying out its functions. 2. Increased efficiency. By having specialized organelles, a cell can carry out complex processes more efficiently than if these processes were performed by the cell as a whole. 3. Facilitation of communication. Organelles can communicate with one another to coordinate the functions of the cell, enabling the cell to respond to changes in its environment and to perform complex tasks. 4. Maintenance of cellular compartmentalization. Organelles create a system of compartments within the cell, which helps to keep different cellular processes and substances separate and organized. 5. Increased metabolic capacity. With a variety of organelles performing different functions, eukaryotic cells have a greater metabolic capacity than prokaryotic cells, allowing them to carry out a wider range of cellular processes. What are the demerits of organelles? The presence of organelles in eukaryotic cells has some disadvantages as well, including 1. Complexity. The presence of multiple organelles increases the complexity of the cell and can make it more difficult for the cell to respond quickly to changes in its environment. 2. Increased energy demand. The maintenance and operation of organelles requires energy, which can put a strain on the cell's energy resources. 3. Vulnerability to damage. The presence of multiple organelles also increases the potential for damage or dysfunction within the cell. 4. Difficulty in controlling metabolic processes. With so many different organelles involved in cellular processes, it can be difficult for the cell to regulate these processes and maintain a proper balance of cellular activity. 5. Costly in terms of molecular machinery. The creation and maintenance of organelles requires a significant investment of molecular machinery and resources, which can be costly for the cell. History of organelles. The discovery and study of organelles have a long and rich history in the field of biology. Here are some key milestones. 1. 1665, Robert Hooke first observed cells in cork under a microscope, which marked the beginning of cell biology. 2. 1838, Matthias Schleiden and Theodor Schwann proposed the cell theory, stating that all living organisms are composed of cells and that cells are the basic unit of life. 3. 1855, Rudolf Virchow proposed the concept of cell division and the idea that new cells are formed from existing cells. 4. 1866, Anton van Leeuwenhoek discovered that cells contain small, granular structures that he called spermatozoids. 5. 1898, Camillo Golgi discovered and named the Golgi apparatus, which was later found to be involved in the modification and transport of proteins and lipids. 6. 1900, Richard Altman discovered the endoplasmic reticulum and Golgi apparatus. 7. 1930s, the electron microscope was invented, allowing scientists to observe the detailed structures of cells and organelles for the first time. 8. 1960s, the concept of organelles as specialized compartments within cells became widely accepted, and the functions of many organelles were discovered and studied in detail. 9. Today, the study of organelles continues to be a vibrant area of research, with new discoveries constantly being made about their functions, interactions, and evolution. What happens if organelles collide with other organelles? If organelles collide with each other, the outcome can depend on the specific organelles involved and the circumstances of the collision. Here are a few possible scenarios. 1. Membrane fusion. If the collision involves two organelles with similar membranes, the membranes may fuse, allowing the contents of the organelles to mix. 1. This can result in the formation of a larger, hybrid organelle or in the transfer of material from one organelle to another. 2. Physical disruption. If the collision is forceful enough, the organelles can physically disrupt each other, leading to the release of their contents into the cytoplasm. 2. This can result in the loss of important cellular components and may damage the cell. 3. Regulation of cellular processes. In some cases, collisions between organelles can serve as a regulatory mechanism, controlling the activity or localization of cellular components. 3. For example, collisions between endosomes and lysosomes can lead to the fusion of these organelles and the degradation of their contents. Overall, the outcome of organelle collisions can have important consequences for the cell's health and function, and scientists are still working to understand the full range of outcomes and the underlying mechanisms involved. What is the actual size of organelles? The actual size of organelles varies depending on the specific organelle and the type of cell in which it is found. Here are some approximate size ranges for some common organelles. 1. Mitochondria, 0.5 to 1.0 micrometers, μm, in diameter, several micrometers in length. 2. Ribosomes, 20 to 25 nanometers, nanometer, in diameter. 3. Endoplasmic reticulum, 0.5 to 1.0 micrometers in diameter, several micrometers in length. 4. Golgi apparatus, 0.5 to 1.0 micrometers in diameter, several micrometers in length. 5. Nucleus, 5 to 10 micrometers in diameter. 6. Lysosomes, 0.5 to 1.0 micrometers in diameter. 7. Vacuoles, 1 to 100 micrometers in diameter, depending on the size of the cell and the type of vacuole. 8. Chloroplasts, 5 to 10 micrometers in diameter, several micrometers in length, found in plant cells only. These are general ranges, and the actual size of organelles can vary depending on many factors, including the specific cell type, its metabolic state, and the stage of the cell cycle. Conclude. Organelles are specialized compartments within eukaryotic cells that carry out specific functions. They play an important role in the structure and function of cells, allowing cells to perform a wide range of metabolic processes and respond to changes in their environment. The size of organelles varies, ranging from a few nanometers to several micrometers in diameter or length. Some common organelles include mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, nucleus, lysosomes, vacuoles, and chloroplasts. The presence of organelles has both advantages and disadvantages for cells, including increased complexity, increased energy demand, vulnerability to damage, difficulty in controlling metabolic processes, and a costly investment of molecular machinery. The study of organelles has a long and rich history in the field of biology, and continues to be an active area of research today. Collisions between organelles can have a range of outcomes, including membrane fusion, physical disruption, and regulation of cellular processes. For more videos, stay tuned with us.